Hello, welcome to Biology, the science study of life. Osmoregulation and excretion, a balancing act. The physiological systems of animals, from cells and tissues to organs and organ systems, operate within a fluid environment. For such systems to function properly, this environment, particularly the relative concentrations of water and solutes, must be maintained within fairly narrow limits, often in the face of strong challenges from an animal external environment. For example, freshwater animals which live in an external environment that threatens to flood and dilate their body fluids show adaptations that reduce water uptake, conserve solutes, and absorb salts from their surroundings. At the other extreme, desert and marine animals face desiccating environments with the potential to quickly deplete the animal's body water. Success in such environments depends on conservation of water, and for marine bony fishes, elimination of excess salts. At the same time, metabolism presents organisms with the problem of waste disposal. The breakdown of proteins and the nucleic acids is particularly problematic since the primary metabolic waste products produced ammonia is very toxic. Studies of how animals meet these physiological challenges provide some of the most prominent. The osmoregulation, which means how animals regulate solute concentrations and balance the gain and loss of water, and excretion, how animals get rid of the nitrogen containing waste products of metabolism. Osmoregulation balances the uptake and loss of water and solutes. Just as thermoregulation depends on balancing heat, loss and gain. An animal's ability to regulate the chemical composition of its body fluids depends on balancing the uptake and loss of water and solutes. This osmoregulation is based largely on controlled movement of solutes between internal fluids and the external environment. The process also regulates the movement of water, which follows solutes by osmosis. An animal must also remove various metabolic waste products before they accumulate to harmful levels. Osmosis. All animals, regardless of phylogeny, habitat, or type of waste produced, face the same central problem of osmoregulation over time. The rates of water uptake and loss must be balanced. Lacking cell walls, animal cells swell and burst. If there is a continuous net uptake of water, shrivel and die if there is a substantial net loss of water. Water enters and leaves cells by osmosis. A special case of diffusion is the movement of water across a selectively permeable membrane. It occurs whenever two solutions separated by the membrane differ in osmotic pressure or osmolarity. Total solute concentration expressed as molarity or moles of solute per liter of solution, or the unit of measurement of osmolarity used in this chapter is milliosmol or millosmol or moles. Millosmoles per liter, which means MOSM each liter. 1 MOSM each liter is equivalent to a total solute concentration of 10 to the power minus 3 m. The osmolarity of a human blood is about 300 mSOM each liter, while seawater has an osmolarity of about 10,000 mSOM each liter. If two solutions separated by a selectively permeable membrane have the same osmolarity, they are said to be isosmotic. There is no net movement of water by osmosis between osmotic solutions. Although water molecules are continually crossing the membrane, they do so at equal rates in both directions. When two solutions differ in osmolarity, the one with the greater concentration of solutes is said to be hyperosmotic, and the more dilute solution is said to be hyposmotic. Water follows by osmosis from a hyposmotic solution to a hyperosmotic one. Osmosis challenges. There are two basic solutions to the problem of balancing water gain with water loss. One available only to marine animals is to be osmotic to the surroundings. Such an animal which does not actively adjust its internal osmolarity is known as osmoconformers, because an osmoconformer's internal osmolarity is the same as that of its environment. There is no tendency to gain or lose water. Osmoconformers often live in water that has a very stable composition and hence have a very constant internal osmolarity. In contrast, an osmoregulator is an animal that must control its internal osmolarity because its body fluids are not osmotic with the outside environment. An osmoregulator must discharge excess water if it lives in an hypoosmotic environment, or take in water to offset osmotic loss if it inhabits a hyperosmotic environment. Osmoregulation enables animals to live in environments that are inhabitable for osmoconformers, such as freshwater and terrestrial habitats. It also allows many marine animals to maintain internal osmolarities different from that of seawater. Whenever animals maintain an osmolarity difference between the body and the external environment, osmoregulation has an energy cost, because diffusion tends to equalize concentrations in a system. Osmoregulators must expend energy to maintain the osmotic gradients that cause water to move in or out. They do so by using active transport to manipulate solution or solute concentrations in their body fluids. The energy cost of osmoregulation depends on how different an animal's osmolarity is from its surroundings, how easily water and solutes can move across the animal's surface, and how much work is required to pump solutes across the membrane because of the difference in solute concentration between their body fluids. Freshwater, for instance, 2,420 mosmo each liter, and freshwater 0.5 to 15 mosmo each liter, and seawater approximately 1,000 mosmo each liter. Osmoregulation accounts for nearly 5% of the resting metabolic rate of many marine and freshwater bony fishes. For brine shrimp, small crustaceans that live in Great Salt Lake and in other extremely salty lakes, the gradients between internal and external osmolarities is very large, and the cost of osmoregulation is correspondingly high, as much as 30% of the resting metabolic rate. In contrast, marine osmoconformers, which are osmotic with seawater, expend little energy on osmoregulation. Most animals, whether osmoconformers or osmoregulators, cannot tolerate substantial changes in external osmolarity and are said to be cytiohalite, from the Greek stones narrow, and hyaline refers to salt. In contrast, uryhyaline Animals also, which include both certain osmoconformers and certain osmoregulators, can survive large fluctuations in external osmolarity. Familiar examples of euryhaline osmoregulators are the various species of salmon. A more extreme example is a fish called tilapia, a native of Africa, widely grown on fish farms for human food, which can adjust to any salt concentration between freshwater and 2000 mosmol each liter, twice that of seawater. Notice that this is the Mozambique tilapia euryhaline osmo extreme euryhaline osmoregulator. Notice the osmoregulation in marine and freshwater bonifish, a comparison between two types. Osmoregulation in seawater fish. Notice the gain of water and salt ions from food and by drinking seawater. Notice also osmotic water loss through gels and other parts of body surface. And notice also excretion of salt ions from gels, and also excretion of salt ions and small amounts of water in scanty urine from kidneys. Another osmoregulation in a fresh water fish. This is a saltwater fish and this is fresh water. Notice uptake of water and some ions in food, uptake of salt ions by gels, and this osmotic water gain through gels and other parts of body surface, and excretion of large amounts of water and dilates urine from kidneys. This is the comparison. Okay, thanks.